As we have learned that neurotransmitters are released by presynaptic neuron, and then these neurotransmitter bind with the neurotransmitter receptor that are present in the postsynaptic target cell membrane. Now, when this neurotransmitter bind with the receptor opens and through this ions flow, and by this way communication or signal transmission occur. Now, in this video, we are going to learn about factors responsible for the duration of neurotransmitter signal. The first factor is the amount of neurotransmitter have been stored and released by the presynaptic neuron. The second is the frequency of action potentials arriving at the synapse. Now, frequency means how many times action potential or repetitive firing of action potentials arrives at the synapse. Now, if the number of action potential increases, what will happen? The amount of neurotransmitter that has been released by the presynaptic neurons also increases, and that also increases the duration of the signaling. The third factor is how rapidly any unbound neurotransmitter means those neurotransmitter do not bind with the neurotransmitter receptor is degraded in the synaptic cleft. Now, synaptic cleft is the narrow space between the presynaptic and postsynaptic neuron. Examples like acetylcholine is degraded by acetylcholinesterase enzyme that are present in the synaptic cleft. Fourth point is how rapidly neurotransmitter reuptake is done by a very specific transporter called sodium-linked Symporter. In my transport lecture series, I have mentioned that Symporters are like a transporters by which two molecules actually moves in the same directions. Here, the sodium and the neurotransmitter will move inside the presynaptic cells simultaneously. Now, let's see some examples of sodium-linked Symporter that are present in the presynaptic axon terminals. In case of acetylcholine, the acetylcholine neurotransmitter is degraded by acetylcholinesterase, and then it becomes acetate and choline. This choline is actually reuptake by the presynaptic terminal, and this occurs by a sodium-linked Symporter called choline transporter. So by this sodium-linked transporter, sodium and choline together come inside the presynaptic terminal. In case of glutamate reuptake, there is two ways. One is direct, another is indirect. In case of direct reuptake, there is a transporter called excitatory amino acid transporter, as because this glutamate neurotransmitter acts as excitatory neurotransmitter, and glutamate is also an amino acid. So that's why this sodium-linked Symporters are called excitatory amino acid transporter. Now, this sodium-linked Symporter is present in the presynaptic terminal. By this way, direct reuptake occur of this glutamate. Also, this transporter or sodium-linked Symporter is present on a glial cells. By this way, the glutamate enters inside the glial cells, and then this glutamate is converted into glutamine, and then this glutamine is actually released by another sodium-linked Symporter called SN1 or system N transporter 1. Now, the glutamine is actually received by another sodium-linked Symporter called SAT2 system A transporter 2. So this is a bypass route in which the glutamate enters inside the glial cells and then released as a glutamine. In case of GABA neurotransmitter, GABA means gamma aminobutyric acid, there is a sodium-linked Symporters that are present in the presynaptic terminal and also in glial cells called GABA transporter, or in short GAT. These are also sodium-linked Symporter. In case of glycine, there is a sodium-linked Symporter called glycine transporter. Now, in case of dopamine, this is called dopamine transporter. In case of norepinephrine, it is called norepinephrine transporter or NET. And in case of serotonin, it's called serotonin transporter or ACRT. These are all sodium-linked Symporter.